Welcome everyone to the AIAA Space Resources webinar series. This webinar series is a publicly available education and outreach oriented webinar oriented at getting professionals, K through 12 students, anyone interested in space exploration and space resources at all, familiarized with the various topics and goings on in our field. And one thing that we have really not addressed in this webinar series yet is agriculture on extraterrestrial bodies. So today we have Dr. Laura Fackrell of Rhodium Scientific and Dr. Rafael Louriero of Winston-Salem State University here to talk to us about some of the challenges and ongoing work in space-based agriculture. With that, I'll turn it over to you guys. I'll be here for anything you need, so take it away.
Awesome. I'm going to go ahead and share my screen so you can see my slides. Does that look good?
They look good. If you want to switch to presenter mode, that would be great, but I will officially leave that up to you.
Okay. Yeah, I'm going to start from the...
Very pretty.
Okay. Are you seeing the slides or are you seeing the presenter?
I am still seeing the sidebar where all the slideshow is still.
Okay. It's sharing the wrong screen. So let me share real quick the other screen. I have too many monitors.
Oh, that's not a bad problem to have, though.
Now I should see the problem. Yes. Okay.
Yes, perfect.
Awesome. Great. So Rafael and I are going to kind of split this up. I'm going to start and I'm going to talk about more on the challenge side and maybe some shifts in perspective. And then I'll turn it over to Rafael to kind of dive into some other aspects of it. But in this part of this, I'm going to specifically be focusing on the role of regolith. But I'm excited to talk about off-world agriculture and kind of the challenges and the perspectives and different ways that we can approach it is one of my favorite topics. So I'm excited to dive into this today. And I'm going to be highlighting a lot of different things that I've covered from multiple areas from different projects that I've worked on. And so there's a lot of people that are involved in that. So I did want to acknowledge that first, just to make sure that science doesn't happen in isolation. We all work together. And so this is just highlights from a lot of work that has taken place in a lot of places with a lot of people. And so I'm just wanted to make sure to acknowledge their contribution to this work as well. But just an outline of what my focus is, and that is I'm going to dive into really like the conditions and characteristics of some of the challenges that we face when we're looking at what are the resources available to do agriculture on Mars, in particular regolith. What resources that present and what challenges does that present, and kind of give that overall concept and like take a look at that, and maybe for at least in some cases, look at a little bit of a paradigm shift in how we kind of focus on that. And then how do we actually conduct that research? We can't access regolith here on Earth. We have very limited access to actual lunar or Mars or other regoliths. And so how do we mimic that and how do we approach that? And then I'll highlight just a little bit on like some of what we've seen and how the plants are responding, how microbes are responding to some of these research projects that we're doing. But I'll leave most of that for Rafael. He'll talk about some of like what are the solutions that we're finding for that. And I'll highlight some of the industry resources that are out there. Having just started at Rhodium Scientific in this last year, I'm learning a lot about the resources and the industry side of this realm of knowledge. And so there's a lot, and so I'll highlight a lot of the opportunities and the facilities and things that we have available there as well. But let's move into the main point of the main topic, and that is looking at the challenges and the potential of regolith simulants in this world of off-world agriculture. And so I like to start off with just this little infographic from NASA that they put together that looks at how do you grow plants in with Mars soil. And I think it's a useful touch point in the sense that it does look at a really important aspect of what plants are looking for, and that's nutrients. Part of what one of the reasons to use a soil or a soil-like material would be because it can provide some of the nutrients that plants would need to grow. And Mars does provide some potential in the sense that it has many of the macro and micronutrients available for that plants do typically use are present in those materials. And you can do a similar chart for the moon. It has a slightly different profile, but you could do a similar thing where you say what nutrients are present in the lunar materials for that. And so this is things that have been detected in the Mars soil or in Martian meteorites at some level. However, I think it's also kind of a limited understanding of what that means. And so I like to then contrast this with a different infograph, looking at what are these conditions. And so we can understand that there are nutrients detected, and so that's a really beneficial thing. But there's also a lot of, I know there's other materials like phthalosilicates or things that could be very useful for agriculture, but there's a lot that we don't know, and a lot of things that may limit that potential as well. And so looking at really the condition of that soil in a broader sense and trying to understand like, are those nutrients are present? Are they available? Are they mobile enough the plant can that in a way that the plant can actually uptake them, or are they stuck in the soil in a way that the plant can't get to? Are there other toxins and things present, that things that are at low enough concentrations that they're difficult to detect with the instruments that we have, but would still be high enough concentration to be of concern? And then there's things like, as particular salinity is one that comes up, and the presence of perchlorates is part of that salinity profile. Is a major problem that there's present in most Martian surface materials. There have been places where perchlorates have not been detected yet, but there are, most samples do have perchlorates detected at some concentration. And even if not perchlorates, then salinity is pretty ubiquitous on the Mars surface. And so there's just a lot of different things that make agriculture very challenging. And so I think this is kind of like that contrasted picture that you can kind of take and understand, hey, look at all these challenges and look at these things that we have to overcome. It kind of then flips the picture and makes it seem like, oh, maybe we can't. But I think that's also a limited viewpoint to an extent. And so I think like this and this present just like these two end members, so to speak, of a picture that could be maybe shifted a little bit. And so I like to actually have a little bit of a paradigm shift and kind of how I've looked at how we do agriculture on Mars, and try to understand this, not just from like, can we use it, can we not? In the sense of, can we just take regolith and put a plant in it and it'll grow and be thrive and be successful? But trying to understand this from a broader kind of system level perspective. And so a really useful framework that I like to use for this is actually the critical zone framework. And so if you're not familiar with critical zone, the critical zone, as pictured here on the left, is typically defined as the upper atmosphere, about where the top of the vegetation will stop, down into the bedrock, where meteoric water or like where there's active weathering taking place in the bedrock. And so in that zone is usually typically called the critical zone. And it's termed this because the critical zone, that zone within Earth's those layers of Earth, is where a lot of the processes and products that interact that make life possible. Where soil formation to all the geochemical cycles that happen within the soil and in these structural, like all these things that are happening that help to support life happen in this critical zone. And the balance of those and looking at these as a whole system and trying to, and they're highly complex and they're highly integrated and they're just so many overlapping pieces to this puzzle that it's really difficult to understand how the system works without understanding the system as a whole. And so we usually dive in and like we understand a specific part of the system, but then we have to come back out and understand how that connects back to the broader system. Otherwise, we're missing a huge part of the picture and how we understand things. And so that's kind of the mindset and the framework of critical zone science that helps to understand how do we interlink these multiple disciplines and these expertise and these tools that we have to really understand the system as a whole so that we can get a broader picture for how things are communicating and interacting in this zone that help us to support life. And so there is a way that we can contribute that to Mars, whether or not Mars has hosted life in the past. There is plenty of effort to make it host life in the future. And even with or without the absence of life, there's still this interactive and intersected various things that interact to create this complex set of systems that work together to create this whole package. And so you can apply a similar concept to Mars, and you could extend that to the moon in the sense of, again, we're wanting to put more life onto the moon, even if it doesn't host life currently. And so this concept of how these systems interact can be really important. And I think it's a useful framework for how we look at off-world agriculture, in the sense that it's not just, like, agriculture is one part of a much bigger system, and there's so many different pieces where these things interact. And so I think that kind of, and if we focus in on regolith as a part of that picture, this continues that framework when we're at trying to understand how we can use regolith as a resource. So in agriculture specifically, if we want to look at a soil and we're understanding that what a soil, the makeup of that soil, where you have like the minerals and the air that are in balance with the water in the soil, and there's usually a good amount of organic matter that helps to improve different aspects of the soil, make it more arable, make it more suitable for growing plants. And then there's this really important role that the microbes play. Even though they're a small weight percent, they make up a huge diversity and one of the most diverse ecosystems out there of microorganisms in the soil ecosystems. And so the soil microbiome is a hugely complex and important feature that we often examine there. And so we have this picture of how soils have grown on Earth, and it is the material in which plants have evolved to grow in. And so these really intricate and interesting and complex relationships between the soil environment and the plant's health and the microbiome that lives in both, and how they interact together is a huge area of research and thing that we're just beginning to scratch the surface. And so if we extend what we know to Mars and try to understand it in a similar framework, we suddenly change this dynamic quite a bit. There is organic matter technically in Mars soil, although it's only trace, and so it's not really in the form that we in the same kind of humic and fumic and other organic components that we usually typically think of as in soil. It's just trace organics in the same sense that we find in a lot of different astro materials. And the water is not something it's frozen where it's present, and it's not necessarily pure water. So there's a lot of those are present, but they're not present in the right forms. And so there's some processing and some interesting things to have there. But you do have this mineral and you have this air. So you have these other components. And so how do we take what's there and restore that system? And so that's one of the things that we can look at and use this kind of framework of like a system-wide framework to look at how are we restoring that system as a whole, and how do we create something that's functionally the same or provides that same functionality. And so that's and restore that into there. And so that's a lot of what this regolith-based agriculture research does. And the way that's useful, and I think why this framework is really key, is because it allows us to kind of shift from this mindset of looking at these huge challenges that make it, oh, we can't even use the regolith because there's too many challenges to overcome with it, to understanding like, well, let's look how we're harnessing it and where we fit it into the system. And this is not just about overcoming challenges, but it's about maybe that particular feature is actually really useful in another aspect of the overall system of trying to create a habitat on the moon or on Mars. And you have these nutrients detected, and so maybe you're going to extract them, and maybe there's materials that you can extract for other purposes that then link into other aspects that link back to the agriculture. I think there is quite a few different people that have done different iterations of how this could look. Just to give a couple of examples from the literature that are out there, where you have you're extracting specific things like mineral nutrients from the regolith, and maybe the leftovers from that then are a useful medium for them carrying over to other aspects. And I think I like the there's a lot here that that I liked, where you like the regolith is input into the system and it's part of this whole process. And there's an integrated system. So it's not just about, are we going to use regolith or are we going to use hydroponics? It's like, we're probably going to use both, or we're going to use different aspects of both. There's materials from all of this that can be used and integrated into a system as a whole. And there's lots of different ways that can be input into that. But it's really complicated. And there's a lot of studies and a lot of research that needs to be done in order to do this successfully and sustainably. And so I think that's the framework that I really like to approach this kind of information with, and to understand off-road agriculture and some of the challenges there are and the opportunities there are in order to overcome those challenges. I'll let Rafael kind of dive into some of the more specifics of some specifics that we're doing in this kind of realm. But I think one of the important tools that we have in this research are simulants. We don't have access to regular simulants here on Earth. For the most part, we have some lunar and some asteroid now, but they're not in large volumes, not large enough to be effective for doing most agricultural studies where you need pounds and pounds of growing medium to do much research. And so simulants are really the bread and butter for what we have to use in order to do the research that helps us to prepare for the real stuff. And so just to give a brief history of what simulants are out there, there's quite a few, especially now that it's growing, that list continues to grow. But for Mars specifically, the original two main ones that existed are from the JFC Mars 1. And so that was from Allen et al. In 1998 that was developed. It's from a pelagitic ash tuff that was ground up and put into it. So basically, And the geologist in me doesn't like to say this, but it's roughly basaltic, and speaking in a very broad sense. And then you have this material, and it was selected because this material had similar spectral properties to some areas of Mars. And so if you do the reflectance spectrum, it had a similar profile in some cases for certain dusty regions. And so that was selected to kind of be that first broadly used simulant. However, there was a lot of issues with the characteristics where there were some things that were not behaving as we would expect them to, and as we've seen them behave. And so like there were some particular issues that caused them to develop another simulant pretty early on, which was the Mars Mojave simulant. And this one was more geotechnical. So some of the features that were problematic with the first simulant that were not problematic in a way that was relevant to Mars. So they're just problematic in general. They developed the other simulant to kind of have another option that was more useful for certain geotechnical applications. However, it also still lacked some certain things like certain salts like sulfates or perchlorates or other minerals and clays and things that we do find in different materials on the Mars surface. Some of those in regolith and some of those in different bedrock resources. And so they're both good starting points for us to get started, and they've been used in a lot of really valuable research. But one of the challenges that we have is then the more we learn about the surface and the more specific studies we do, we need to be able to target more specific characteristics that aren't reflected in those initial early simulants. And so there's been a big push in recent years to kind of create additional simulants that really target more specific characteristics and target different applications. And so there's the Mars Global Simulant that's really commonly used as well now, that try to reflect a lot more of that, some of the other components that aren't typically found and have a more accurate mineralogy to Mars, and try to match that more precisely. There's simulants that were developed by during my station that we try to develop for specifically for agriculture, and so really trying to target different characteristics important for that. And so we developed several simulants for that, and we did some studies with those. And there's several more that have started to come out that really try to target these different features, whether it's iron extractability or looking at making sure that you're mimicking the right characteristic for the right application. And so that's a really important aspect that I think is for simulants, and I'm glad for the development of so many more, because it gives us greater knowledge and greater breadth of things that we can test and to be more robust in our results for what is actually happening. And so this brings us to like, here there are some highlights for like what we've been able to do. And so a lot of the classical and like the first research that was done to do regolith-based research with agriculture is that you stick a plant in a simulant and you grow it and you see what happens. And I think that's really important for the groundwork and like laying that foundation. And so we'd have some of those results where a lot of the studies kind of approach from this particular approach where they see how the plants are responding to these simulants. So some of these original ones do use this particular one, they use the JSC simulant, which is probably not the best simulant to use for agricultural applications, but it does give us that starting point and a where to start from. And so that's some information that those are there. With the new MGS simulant, there's been additional studies that have kind of looked at various simulants and seeing that there's a wide difference in how plants respond to different simulants. And so you want to make sure you're selecting a simulant where the responses are going to be relevant because the characteristics mimic those that are important for plants in agriculture, and the characteristics that they mimic most accurately. And then also, and we saw the same thing in the simulants that we were using out in different simulants had very different responses. So various materials on the Mars surface are going to respond very differently. And it's not a simple just stick it in and see if it grows. It's a much more nuanced. There's a lot more work and a lot more challenges and a lot more possibilities to explore and to like look at different solutions and how they integrate together. And so there's a lot of information, a lot of work to be done there. And some of that work links back to the microbiome. And so there a lot of the studies that we looked at, we were looking at specific like salinity. This particular study looked at salinity with Mars and looking at how plants respond to Mars like salinity. And if you inoculate them with certain microorganisms, does that improve their response? And it did. There was actually a pretty significant difference between the plants that were and were not inoculated in both the lower and higher salinity stresses that we looked at for Mars. And so the microbes gave a lot of help. And so I think Raphael will talk a lot more about those kind of things, but that's one of the solutions that we've seen henceforth. So there's a lot of opportunity there for research to be done. We also see, but there's also cautions for that same feature of the microbiome is that not all microbes are beneficial. And some of the work that we were looking at when we were recycling the waste and trying to understand how the waste was recycling, there are pathogens present like Salmonella or E. coli in these recycled products. And so you have to be also cautious of like what the microbiome you're inviting in is the microbiome that you want. And so there's a lot more complicated. You can't to really understand how that microbiome is going to respond and all the integrated things there. And so there's a couple of highlights for that side of things, and how the microbiome is an essential part of that picture. But I think that kind of brings that circle to where I'm about. I'll transfer over to Rafael. But I do want to highlight some of the things that we're doing. So I just started with Rhodium Scientific in this last year, and so I've been with them several months now. Just kind of understanding the industry side of this burst. I've transferred recently from the agricultural industry. And there's been a lot of really amazing things that we've been able to see. We're looking at these biotech advances and applying them to space. There are some huge, amazing biotech that has been going on that is applied every day in the industry on Earth. And so how do we apply that to space? And so that's something that Rhodium has excelled at, and that I'm really excited for all the things that are available. And these are just some of the different chambers and the facilities that we have that we've used for several of our agricultural studies before, and there's more to come. So that's part of my job, is to like expand that and to lead our agricultural department. And so that's what we're applying. And so some of these different chambers and these different things that we have available is just shows the opportunities that there are in industry to do a lot of this as well. And so that's something that not everybody is always familiar with, is that what is available from the industry side of biotech and how is that being used in space. And so this is just to kind of highlight some of that and some of the missions that we have and some of the facilities, including some of our little plant chamber, Sophia, that's able to like do a lot of reiterations and get the higher repetition and manifest on these smaller plant studies. And it's a little bit easier to use. And so that's just one of the experiments that's happened for that. And so that was pretty exciting to like just get some of these results for understanding how the plants are responding to the space environment and some of the challenges there. But I don't want to take up all the time. I want to make sure to save plenty of time for Rafael. So I'll go ahead and finish up my side of it. If you have questions, put them in the Q&A chat and we'll get to them at the end of the talk. We'll have time for some Q&A. And I'll also try to keep track of that, but we'll turn the time over to Rafael and let him kind of take on from here.
Thank you, Laura. I will share my screen now.
Yes. Yep.
Do this. Okay. Can you guys see my screen okay?
Yes, the slides look great.
All right, awesome. So, well, this is me. If you didn't know who I was, you wouldn't be here. So the intent today is for me to talk a little bit about things that we have been doing here in the lab regarding trying to solve the problems that Laura just put in front of you. Laura has set a scenario, a very gruesome scenario, difficult scenario for me to dive in. But as Laura Laura and the lab have been collaborating on a number of projects. It is something that we're trying to do to make this work. Basically, off-world agriculture is us trying to hack agriculture once again. For us as humans trying to understand how agriculture is going to work somewhere else, right? A lot of the conditions, pun intended, is completely alien for the plants to deal with, and it's our job to try to make these plants a little bit more comfortable. Over the past 5 years, we have been focused on agricultural, regolith-based agriculture, mostly focused on the lunar side of things. But we have also been dipping ourselves into the Martian side. So the lab is myself and a bunch of students. And as you guys know, students commit mistakes, but some of these mistakes are actually very good when it comes to science. A lot of good science came from good mistakes that have been committed by not only by students, but by the researchers themselves. When we first started doing this, we used to completely sterilize regolith and just to see the responses that the plants will have. I'm a plant physiologist. I like to analyze how the plant is feeling, and through phytohormonal analysis, and try to understand how we can potentially change that. By completely sterilizing the regolith, not allowing any kind of pathogen there or any kind of bacteria to begin with, we started to see that the plants would not do very well. They would not develop into any kind of fructifying, they wouldn't develop in any fructifying body. They wouldn't form flowers, and they would form flowers. The flowers will fall as in a representation of stress. So we were very frustrated when we started doing this. A lot of tears. But one day, maybe a Friday like today, a student forgot to sterilize the regolith, and we were unaware of this. And we started to grow the plants. And one side of the chamber started to have plants that were very, very, very nice looking. And the other side of the chamber didn't. And what we started to analyze was maybe the CO2 is off on that side of the chamber. Maybe the irradiance is off. Maybe the temperature changes. But no, all the environmental factors that were influencing the plants were fine. And then we started to analyze the regolith to see what was going on. And lo and behold, that regolith was contaminated. And as you guys can see here, we have A and B, and we were growing peppers at the time. As you guys can see, the contaminated pepper showed way more leaves, and it was the biomass production was incredible compared to the ones that were not contaminated. Mind you, we're still able to grow some plants, but they struggled to grow, and usually those plants will not come to full term. Those plants will die eventually. But the plants that were contaminated, they were able to differentiate tissue into inflorescence, and those flowers produce fruit. Mind you, these were supposed to be normal sized peppers, but at the same time, they produce fruit, and those fruit produce seeds, and those seeds were viable, something that before that we were not able to achieve. So we crossed over into trying to understand how we could potentially capture that. Because plants, when they are in the soil or in regolith for all that matter, these plants, what they're going to do, and plants in general, they are going to produce root exudates. Root exudates is a way for the plants to communicate with the microbiome and recruit them to do what plants don't really know how to do or do poorly. So root exudates is an exchange that the plants are going to do with the microbiome around them. So I don't know if you guys play Dungeons and Dragons, but I do, and the lab does. And the idea was to create a 6-sided die, and in each one side of the die, we will put a bacteria that could potentially have plant growth promoting activities, and that we could potentially explore that in different combinations based on the cultivar that we were growing, and also the type of regolith that we wanted to subject those plants to. And that's why how we created the microbial cubes. And these are pictures that are currently being used in Brazil, which is pretty cool, in a collaboration that we have with the lab there. And we're kind of transferring this technology to other places. So how do we do this? How we initially assess the feasibility of purposely contaminating the regolith, but with microbiome that we know the composition of. The computer keeps shifting, I'm sorry. So we have chosen a recipe, a combination of microbes that we could inoculate in these cubes. We called this recipe Lat1 because it was geared towards lettuce. As you guys can see here, I'm not going to share the recipe with you, but as you guys can see, the hardware that we use is very high tech, very high end. There are trays, silicone trays that you can get from Amazon for less than $10. And these are great because you can autoclave them and use them repeatedly. So we started to assess the growth of these individuals that were exposed to this microbial recipe with utilizing the cubes. So basically what you do is you inoculate a pre-inoculated cube with the microbiome that you want, and then you associate the seed with it, and then you put in regolith. as you guys can see, based on the visual results, and then I'll go to the numbers in just a bit, all the contaminated, purposely contaminated plants with the recipe that we have developed started to have a very active root exudate formation and early recruitment of bacteria. And also they displayed very good overall morphology of these individuals. These individuals will grow into term, these individuals will produce inflorescence, and will produce seeds as well, which for us was a major thing for us to kind of crack the code on how to continue to do that without it being a fluke. So these are some of the numbers, as you can see, in terms of edible biomass and total biomass and root exudate yield and the early recruitment of bacteria. Everything was working on our favor. So the purpose of contaminating the regolith is something that we necessarily will have to do to make this work, right? And it's best for us to, it's kind of like jumping out of an airplane. I mean, jumping out of an airplane wearing a parachute is better than not using a parachute. So we're just creating the parachute now, trying to understand what kind of microbiome we have, and that we can inoculate the regolith with without any potential pathogenic ripples that could affect the crew or the plant itself. We have observed that if you guys shift to this graph over here, that has the phytohormones there, you see that the ones that are labeled MR21, which was microbial recipe 21 days, you guys can see a spike in one phytohormone that's called salicylic acid. Salicylic acid is the phytohormone that is going to allow plants to fend off pathogens, which for us was a huge surprise because our microbial recipe, this specific one, LET1, will create some sort of a vaccine-like effect to plants and allowing the plants to be free from pathogens and free from any kind of pathogenic influence. Also, some tests that we have been conducting, and as Laura pointed out earlier, what we wanted to know is that if any pathogenic bacteria or fungi will come around the rooting zone of that plant that has been previously inoculated with our recipe, would that pathogen kind of sneak in and take the plant over? And what we have observed is that once you have that pre-established microbiome already associated with the plant from the get-go, there is very little to no chance for that plant to be infected, which is awesome, right? And so anything that happens around the plant, the bulk regolith area, cannot affect the microbial community that has been formed around the rooting zone. Right now, we're trying to expand our microbial cube endeavors, trying to put a cube within a cube. So trying to understand how we can put a little bit of water and nutrients around the microbial cube and the seed to try to associate that with long-term space travel. So if we inoculate these cubes here on Earth and put them in the spaceship and we're taking them to Mars, during the journey, these cubes would kind of grow the plant, right? And we will do a system of plug and play. You would just take those plants out and just plug into the regolith, knowing that the microbiome has already been established during the time that you're traveling. The problem is, and there is always a problem, the current technology that we have only sustained the plant for 21 days. We're still kind of hammering together, trying to understand how we can make this better and make the plants feel a little bit more comfortable for a longer period of time. But we're getting there. Talking about a longer period of time, after we fully understood, not fully, but we had a pretty good idea how the plants responded to the only regolith stress, we trying to put an additional stress in these poor plants, trying to understand how partial gravity would influence the plants when associated with regolith. This was something that was done with one student in the lab, Taylor Johnson, and the results were pretty interesting. And when it comes down to the overall growth of the plants, Martian regolith usually is the best route to go, especially when it comes to the micro and micronutrients that are available and the bioavailability of those in Martian regolith compared to lunar regolith. But as you guys can see here, with the overall distribution of plant hormones there, usually you're going to have spikes in gibberellin, which is GA4, and other hormones that are going to be responsible for root formation and early germination. So, taking a deeper dive into it, we observe that individuals that have been exposed to lunar regolith will create more lateral roots. And that is very good. First of all, because in partial gravity, these individuals can fully attach themselves into the substrate. Also, not that they were going to fly away, but it just the plant feels a little bit more comfortable when it comes to that. And also, these individuals save a lot of water. So if you have a lot of lateral roots, you have a lot of roots that are going to be on the upper strata of the regolith or soil, and these individuals are going to consume less water and produce the same amount of biomass, which for us was always a struggle for us to have a good biomass production with whatever we had in regolith. So what was influencing there, we saw that the levels of calcium in lunar regolith were slightly elevated when compared to Mars. So we started to try to understand how this could potentially influence root formation, and it does. So the root formation is influenced by a hormone called auxin, and the pathway from formation of auxin is usually mediated by calcium ions. And and this gibberellin and auxin signaling is going to influence the formation of lateral roots, the elongation of these roots as well. Which currently a student here, Sarah, is evaluating the genetic pathway. This is how this is taking place genetically, because morphologically we can see it, and she's just closing in on the publication that we're going to put out before she leaves to Florence to Technology to conclude to do her master's. So she is just closing up, and we should have this paper out in June or so. So we can fully understand how the levels of certain compositions of regolith is actually influencing the overall root morphology, root architecture, or the overall morphology of these individuals. Another experiment that we have done is try to understand how the granulometry, how the physical properties, and not only the chemical properties of the regolith influence plant growth. Trying to understand if there is some sort of appropriate granulometry in regolith. What is that granulometry? What is that threshold? And can we expand that to every single species, or each species is going to have a specific size of regolith that is interesting for us to use? We have tested a bunch of sizes of particle sizes with lettuce. Lettuce was our main target because we understand a lot about lettuce, especially when it comes to stress. So we have a lot of information that in literature in our lab. And when we got there, we have seen that there is a specific granulometry that regolith is going, the plants are specifically lettuce is going to be very comfortable in. So we started to try to coin the term agricultural grade regolith in terms of trying to put out, and we were talking, I think that Laura kind of dabbed around this, and the ideal area for us to test when it comes to the regolith that is being manufactured, for us to make a request to say, I want this granulometry with this amount, this composition for me to test this specific feature that I'm trying to test. We're trying to do something that has never been done before. It is extremely challenging, so we need to narrow it down and have standardized methods to test everything. And this is a good way to start for us to have like agricultural grade for lettuce, agricultural grade regolith for peppers. So the trying to understand the granulometry, the ideal granulometry for each one of these crops, and see not how the physical and chemical properties are acting at the same time. So if you have the granulometry down, we're just focusing on the chemical properties of the regolith, because you have a jumbled mess of everything happening around the root zone, making the plant highly uncomfortable. So if you can take one of those out, you can focus more on the other, knowing that the other factor is not influencing your results. Trying to also make the plants a little bit more comfortable. We know that NASA, I'm not going to say this like directly, but it's not that it doesn't have interest in regolith agriculture. It's a plan B. So just trying to prove our point that, hey, we can integrate systems, right? If we want a large production of biomass that is going to help the crude that is there, and then an extensive population, if we want that to happen, we need to integrate systems, right? Because if you have just a hydroponic system, it has limitations. It costs more. It's more prone to being affected by pathogens, and also the overall structure is, I mean, it's highly costly for us to put that on the moon or Mars. So, what if we had a full integrated system that we started in hydroponics and then transferred into regolith? So, Sydney Wharton was a student that tested that, and what we could observe is that regardless of the amount of days that those plants were in hydroponic to regolith, they showed an amazing amount of gain in biomass. I'm sorry, gain in biomass, and the responses of the plants overall, regardless also of the species that we have tested, were always positive. So if you start in hydroponics and transfer into regolith, the plant is going to transfer a lot of the microbiome that is in hydroponics as well. And when you put in regolith, some of the microbiome is going to go away, some of them are going to stay around. And if we can associate that with microbial recipes and pre-inoculate these plants in hydroponics, we kind of have the perfect scenario there. However, not all plants respond the same. As you guys can see here, these are radishes that look like carrots. And they are not the size of a regular radish, although they are supposed to be. And was trying to understand what are the genetic triggers that make pithiness happen, like this particular response that radishes have, because we cannot just have Pekingese crops and we cannot just have lettuce. We have to expand our cultivar portfolio there, because it is very important for people to have choices, especially being that time away from Earth and not having a stable diet as well. So having all these options available is important. So we're currently trying to understand what is the phytohormonal play that is happening there, the crosstalk, for us to try to understand how these plants can be a little bit better when it comes to the overall production of the biomass that we want. And on the side there, you can see that now we have come down to radishes looking like radishes, but they're still not the size that they're supposed to be. And this is my life. Usually plants don't look like what they're just supposed to, and then we get there and they taste horrible, and then we work on the taste and move from there. The idea of that is of this research is just that. Can we create a tomato species that is very good for stressful situations? And this is just a breakdown of what we have done, but the pictures are cooler than this. But what I can tell you is that we were able to do so. So over the past 2.5 years, we have been using just traditional crossbreeding techniques to create our own tomato variant, which we're calling ink spot tomato. We use tomatoes from all over the world, mostly from Eastern Europe. And I don't know, I mean, I'm a plant nerd. If you're not part of, I don't know, tomato breeding forums, you should be, is a very interesting place to be. And the exchange of seeds that happened is very interesting. So I was able to get my hand on those. And here in the lab, we cultivated a lot and trying to make it ours in a way, trying to understand what we were getting in terms of cold and drought tolerance variants, and also dwarf tomatoes, and also anthocyanin rich tomatoes. Why did we wanted all that? Cold and drought tolerance is important for transport, and also for the lack of water around these individuals, maybe during transport or maybe during some sort of event that a chamber may not work properly or something like that. And anthocyanin rich, anthocyanin is a flavonoid that is going to be responsible for antioxidant properties, and that is going to allow the plant to be more effective in sustaining any kind of stress. And that's why the fruits are going to be a little bit darker in anthocyanin rich variants. So what we have done over the past 2.5 years is try to breed those and not only make them more resistant, but also make them bioindicators, which is the coolest part of this. These tomatoes, they are going to respond to stress by making the fruit darker. So as you guys can see here, and mind you, we didn't start with this. Some of them died during this process. We had a bunch of variants that didn't make what we wanted them to do, which we still have here in the lab that we keep as pet plants, but as pet seeds. They're not plants yet. So right now, what we have, and this is maybe the coolest thing that we have done so far in the lab, is our very own tomato. So on the right side here, you're going to have just a tiny tomato, just a dwarf tomato that you can buy anywhere. And on the left-hand side, you have ours, which is much darker, and they produce a lot of flowers very early on. And all those flowers are going to become fruits. I mean, most of them. We have like a 90+% rate of flower to fruit ratio. And they're also smaller. The leaf area is smaller, so transpiration doesn't occur as effusively, let's put it this way, compared to the other species. And so you save a lot in water as well, and they're very tolerant to any drought stress. But another cool feature is the fact that these tomatoes are going to respond differently when it comes down to fruits. So here from left to right, you have tomatoes that have not been exposed to stress, mild stress, and complete disaster scenario. As you can see, the tomatoes get darker and darker and darker. The fruit gets darker and darker and darker as the stress increases. And the tomatoes that are produced, they're a little bit bigger than the regular tiny tomatoes that you would have, which is very cool as well. Laura was talking about the importance for us to try to understand the regolith as well and try to integrate that somehow into the work that we do. I mean, I'm not a geologist, Laura is, but I know that we can extract something from the regolith, right? We can get stuff out. So one of the projects that we're running here in the lab is trying to extract components from the regolith and make some sort of a concoction so we can put into a hydroponic system. So we're not directly utilizing the regolith for agricultural purposes, but indirectly we are. So whatever regolith that we can put our hands on, we use some bacteria to leach out whatever nutrients we can from the regolith. Some people have done chemical leaching before, but this is more the costs are increased when you do chemical leaching, and also you have to dispose of those chemicals, and you may not have the ability to do that on the moon. So you're dealing with bacteria here, which is very easy for you to have a bioreactor here and there, just keep them around, and mixed in with several species, we have been able to leach out some of the components that we want from the regolith and use it as nutrients in a hydroponic system, which is pretty cool. And the last thing, maybe the last thing that I have to say, and then I'll be quiet for questions, is PlantTrack. PlantTrack is a project that we have been doing with Dr. Kendall Lynch and Anna Simpson, and had the idea to extract perchlorate from Martian regolith utilizing naturally occurring bacterial consortia. So you will get kind of like a microbial recipe, did some aerobic treatments, anaerobic treatments. There is a group with Anka and Drew from Florida Institute of Technology, and I forgot where Anka is. I think it's University of Arizona. Laura can, yes, maybe, yes. Well, they're doing the same thing, which is awesome because all the results that we can get will help. We get something kind of like a slushy like this, and we try to make it work when it comes to growing plants on it. And what I can say, I cannot put the results out, but what I can say is that we have been very successful on trying to exclude perchlorate from Martian regolith. And we have been able to grow plants in those in that regolith as well. Currently, we have Joy that is taking care of that. And you can see Joy, they are very happy after an hour using this there, trying to manage all the plants that she has growing. And this is an asset that we can potentially apply to also degraded places here on Earth, especially from mining companies and other activities that completely degrade the soil. So we could potentially use the same approach here on Earth. And this is one of the things that I always try to close my talk with, is that people are often ask, why are you growing these things on Mars? Why are you doing this for the moon and Mars? But while a lot of the technology, or if not all the technology that we create for growing plants in these completely inhospitable environments can be transferred here to Earth to help with food security in places that suffer with soil degradation, and also in arid, desertic places, we can help people get access to food. So I will close there, and we'll close with Mr. Spock holding a plant, and I'll open for questions.
Awesome. Thank you both very much. Really cool stuff, and great to have both sides, the regolith perspective and the plant perspective. So yes, with that, let's use our remaining time for questions. I'll start with the first received and work my way down. Cynthia Valenzuela asks, are there any opportunities for internships in Rhodium Scientific?
Make sure to unmute myself. They do have internships at times. I'm not in charge of that part of Rhodium, but I can certainly put you in contact with people who are, if you want to email me, or if I'm not sure my contact information is available on LinkedIn, then you can certainly reach out and we can get you connected.
And you can go through me, Cynthia, however you saw this. If you have access to message me, please. If not, what I will actually do, and Laura, I can punch you. Do you want me to punch your email in the chat?
Yeah, that would be fine. So if they have questions or they just want to learn more information, I can certainly connect them to the people that have the information.
Cool. I'll put that in the chat in a second. I'm going to jump to the next question, and then I'll do that. So Jeff Taylor, hi Jeff, asks, have you thought about the timing of developing agriculture on Mars versus the search for indigenous life on Mars? Bringing terrestrial life to Mars for agriculture could ruin the record of indigenous life on Mars.
This is something I think a lot about. So one of my hats that I wear is space biology, but I also have another hat that I wear that's an astrobiologist. And so I have done a little bit of work there, and so I've thought about that a lot. And I would say it's not just tied to agriculture, like things that we bring for agriculture, just bringing people to Mars. You're bringing bacteria and things with those people because you can't sterilize a person. And so no matter what, as soon as you bring like human exploration to the Mars surface, that's what you're putting at risk. And so I think that's something that COSPAR and like planetary protection and those kind of groups, they think a lot about how we're going to approach that. And so there's a lot of discussions and a lot of thought going into how we time this, at least from NASA and from these international space agencies, of how we do this and how we can maybe create some areas of Mars that are protected areas, that we preserve those for future exploration, and how we balance the needs for planetary protection and for discovery of astrobiology with the needs of space exploration as well. So definitely a really important question, and thank you for tagging that in.
Awesome.
I have nothing to add.
Yeah.
I mean, we were going to contaminate regardless. It's better for us to understand what we're bringing with us first, right? And if we're bringing anything at all. But at the same time, if we are there to stay, either Moon or Mars, but we're eventually going to do that. I mean, we already have on the Moon, so it's, yeah.
Well put, guys. This one was put in before your talk, Raphael, so I'm sure Chris Kelly would agree that your presentation was very enjoyable, but enjoyed that presentation, Laura. All right, then Jason Matthews asks, do you have plans to investigate mixing human-derived fertilizers into the regolith?
So I think there's like I think that's like yes, but it's how much of that do you bring with you on the ship and how much can you extract from what's there? And so those, like, it's not just about like the compost or like microbiomes, it's also about like nitrogen or phosphorus or just technical fertilizers, and I think the ideal is that you want to make as much of that in situ as possible to save on how much you have to bring with you. But there are limitations. There's not a lot of nitrogen on the moon or on Mars. And so like, and how much that can you extract from human waste? So there's going to be a balance of figuring out how much to bring, for sure.
Yeah, I think that the major problem there are any potential pathogens that you can bring in by doing that. There are effective methods in place to do that, but I think in the long run, it's tricky for us to try to make this work. especially but in terms of water utilization that you can recycle. International Space Station, I believe that would recycle like 98% of the water or something like that. I don't know. But when it comes to overall human waste, I'm a bit skeptical when it comes to that for now.
Good to know. Jason Matthews also asks, there are ongoing experiments incorporating mycelium into lunar regolith. Is this something which you have or will investigate?
We have done some experiments with fungi here. The problem is that you have to put organic matter into the regolith and then grow the fungus. Fungus, it's going to need organic matter to create a mycelium and then generate the fructifying body. So it's a process. I think that initially it's interesting for you to potentially try to have some sort of pioneer species that is going to grow, fix nitrogen, and then die and create that bed of organic matter. Then you infuse the fungus that you want to grow, and you could potentially form a fructifying body. I know that Andrew Palmer is, I do believe, a master's student, a PhD student, Andrew Palmer, is working with it at Florida Institute of Technology, trying to grow fungi in Martian regolith.
And the other thing I would add to that is that he talked about you have to input organic matter. Organic matter is definitely one of those things that there's going to be a lot of in waste. And so figuring out various appropriate ways of utilizing that waste so that they're retaken into the system is part of that process. Not all the waste is going to be usable for that. Like there's different parts you're going to have to use for different things, but that's definitely something to, and mycelium, I think, are a really powerful and important potential microbe to include in that microbiome. So certainly part of the picture.
Great. Next question from Jen Blank. Hi, speakers. I'm wondering, one, how does your food production effectiveness correspond to nutrition for humans? And two, what about aerosol delivery of water plus nutrients? Could this complement the hydroponics work?
Laura, do you want to go first?
Sure. So I think, this is directly like food production to nutrition. So the whole point of the food production is to produce nutrition. And so we definitely want to target and like approach the food production in a way that we're focusing and making sure that nutrition is part of that is met. The nutritional needs are met for humans. So that's a big part of that research is transitioning from just like plant science in regolith to crop production regolith is a huge step. And I think that's something that's actively being attempted right now. So like there's a lot of pieces in play that will allow for that transition, but it is a transition that we're still trying to meet. And I think like aerosol delivery, so I'm interpreting that as aeroponics, which is like a variation on hydroponics is what I'm interpreting that as. Feel free to post another question if that's not what you're intending. But yes, I think that's also part of that. So the integrated system where you have different approaches and that you do different things that are appropriate for different types of plants. So you can expand and diversify how the plants are growing. Because a lot of plants are going to do really well in aerosol and aeroponic kind of stuff, but they might be able to do different things with in a hydroponic system, and you might be able to do different things with a more regolith based. And so like try to integrate those systems and like balance all of this together.
Yeah, just to complement, when it comes to nutrition, you need a lot of edible biomass, right?
Mhm.
So whatever you're producing, you need to compensate for the amount of energy that you're putting into the system. The system needs to give you back a lot from what you're putting in. So a lot of edible biomass needs to be there, and also the variations of the cultivars that you have need to form a balanced diet, right? Based on a plant-based diet. Obviously, there are potentially going to be complements there. But those are the key aspects that we are always looking for is what is the plant is producing in terms of overall vitamins or anything, any extra components in terms of human nutrition, and also how much biomass is the plant producing based on the energy that I'm giving the plant to produce something. And the other question was aeroponics. I'm a little bit skeptical when it comes to hydroponics. I prefer hydroponics, and that's a big thing for me to say. Because aeroponics in partial gravity and in microgravity can be tricky because whenever it's being released, the water behaves a little bit differently than what behaves here on Earth. And the water needs to be on the surface of the root so it can be properly absorbed, and that can be a little bit tricky. if you design plants to have a different root architecture that forms some sort of a net-like structure to capture that, that is pretty cool, and that's something that someone can do. But right now, I don't think that aeroponics is something that it's feasible.
Well put. I did not know a lot of that. So, cool. A few more questions left. We are at time. Are you guys cool with blasting through some of these or? Okay. All right, so we'll go quick. Jason Matthews asks, how about GMO plants? Have you investigated or have plans to use modified seeds and investigate potential benefits?
Sure. Yeah. Sure. Yeah. No problems there. Um, I'm not.
It's something that I've explored specifically, but it's definitely a big area for potential. Yeah, go ahead, Rafael.
Yeah, no, I prefer traditional techniques. I think that through the generations that we can see the differences. And the coolest thing is that you can pick and choose whatever you're getting through your process. Obviously, it takes longer, but during this process, we're just connecting the genetics of these individuals that someone could potentially explore. So that's great. But 100%, if that is your goal to completely alter an individual for a determined purpose, yeah, go ahead and do it. I think it's great, and especially when it comes down to being more tolerant to stress. Something that these plants are always going to be subjected to, because once again, throughout the evolutionary history of these individuals, they have never been in partial gravity. So if you can, or microgravity, although we know that plants have been grown in the International Space Station and have been grown on the surface of the moon. Um, uh, there are some alterations that are going to happen here and there. And in order to try to alter that, you can potentially go to the GMO route. It's not what I would do, but go ahead and do it. And I think it is great.
Awesome. Thank you both. Uh, Carlos Boristeros asks, would you be willing to share more detail what are the properties or specific requirements for agricultural grade simulants? I would love to hear more about it via email or similar. I am building a Regulus simulant startup in Europe and would love to hear more. Carlos, if you are still on, which you are, um, I did punch Laura's email in the chat. You should see it in there. And I'm sure Raphael would be happy to comment as well.
Yeah, put my email there. I can send you the ranges for specific variants and cultivars that we have had here growing in the lab. But Laura has made her own regolith before, so she's way more capable of guiding you through it. But I'll happily share with you the ranges for each one of the variants and species that we have done here in the lab.
Awesome. Raphael, thank you. I did just punch in your email in the chat, so folks, you should have his email in the chat as well. If not, and you have a way to contact me, please reach out and I can provide that information. Oh, hold on, answer live. I'm messing up on the interface side here. Okay.
Okay.
Shahryar Sazad, it was a very insightful webinar I am from Bangladesh and very much interested to work in growing plants in lunar soil. Is there any chance to work with Raphael? Well, I punched his email in the chat, so go ahead and reach out. So then there's that one. Olivia Gomez Holzhouse says, great scientific presentations for Raphael and Laura. A lot of relevant research to take us off planet. And then, yes, I did punch Laura's email in, so feel free to keep in touch with both of them. Jason Matthews says, very informative presentation and great answers. Thank you. And Carlos says, great, thank you so much. And that's all I see in the Q&A. No one else, don't you dare punch anything in, because we are over time. And I usually try to be real good about being on time with this. So with that, I will personally say, Raphael and Laura, thank you so very much. No, Jen, we're not doing that. I'm kidding. If if you want to reach out to Raphael and Laura, go ahead and do that. Sorry, done with questions. So guys, thank you very much for your time and sharing your expertise here. This has been a really, really cool presentation. I learned a lot. I think everybody did. And for now, I guess like I said, we're over time. So from the AIAA Space Resources Technical Committee, thank you to our speakers. Thank you all who attended. From me, the same thanks, and I will talk to you all soon.
Thank you, guys.
Thanks.
Bye-bye.
Awesome.
Bye.
Bye, everyone. Have a good one.